Good evening, ladies and gentlemen. Today, we will be discussing ion channels and neural communication. I hope you will enjoy the video. Let's begin. So, let me just give an example that we do every day of a simple neural communication. Well, let's say you would like to move your finger, as we see here in the picture. What happens simply is your brain sends a signal to your hand. Well, today we're going to find out that it's not so simple as we might think. So the literal or basic train of communication is that our motor cortex part of the brain sends a transmission to the nearest neuron, and it goes on to the central nervous system, down the spinal cord, until it reaches the motor neuron that is connected to your hand and eventually to your finger, and then we get our finger to move. So let's begin on how exactly does this happen. First thing that we need to know is the resting membrane potential. What is the resting membrane potential? Simply put, it is the millivoltage difference between inside the cell and outside the cell. Now, I want to explain, rather, how does the inside of the cell get a different voltage than the outside in the cell? Well, let's begin. So, first thing is that the resting membrane potential exists in all cells, meaning that you're right, we're speaking about neurons currently, but we should know generally that every cell in the body has an existing membrane potential between inside and outside the cell. Now, as we've mentioned, the resting membrane potential is the voltage difference across the cell at rest. So, as we're going to learn soon, there's a different voltage difference as the neuron is firing and as it's repolarizing. But we're talking about, when we speak about at rest, when the cell is not moving and not doing any action, then the neuron cell's resting membrane potential is, according to most textbooks, negative 70 millivoltage to negative 90 millivoltage. Now, we'll expand more about these numbers in a minute. The first reason the resting membrane potential is negative compared to the outside is on the membrane of the neuron, as we can see in this interesting diagram. It's not really too specific, but we know from the textbook pictures that these sodium-potassium ATPases carry three sodiums out of the cell from inside the cell, and at the same time they bring in two potassiums from outside the cell and into the cell. Now, if you do some simple calculations, taking three positives out of the cell and only bringing in two positive ions back in, that results in a net positive charge on the outside of the cell, which means it's a little bit more negative inside the cell. So, as an example, let's say we start off our resting membrane potential millivoltage at 0 compared to the outside. With these sodium-potassium ATPases, it becomes negative 5. Okay? Now, the next reason, which is probably the major reason for the negativity inside the cell, is our potassium leaky channels. Now, as we see here, there is more potassium leaky channels than there is sodium-potassium ATPases. And not only that, it happens more frequently. One of the reasons being that sodium potassium ATPases require, as the name inquires, ATP to function. So they require energy. While our potassium leaky channels, as the name again inquires, that it's leaking. It doesn't require energy to actually transfer it out. It just leaks out. It's an open channel. Second reason why it happens more is obviously because there's more of it. There's more channels than there's the ATPases. Now, let's see exactly what's happening here. So, why is potassium leaving the cell? What's wrong with home? I mean, none of us want to leave home. I bet most of us like class at home on Zoom better than class in school, you know? So, if potassium's home is inside the cell, why leave home? Well, there is such a thing called the law of diffusion, which basically means that anything that has a higher concentration will always flow to a place that has lower concentration of that thing. In our case, it is potassium. The amount of potassium inside the cell is extremely high compared to outside the cell. Therefore, the law of diffusion takes place, and naturally, potassium moves outside the cell, slowly but surely. Now, as you can see behind our potassium, there is A-. This refers to negative anions that are inside the cell. There are many different negative anions, small ones like phosphate groups. There is RNA inside the cell. We have proteins that are made up of amino acids, which are negative. So the cell has a bunch of different negative anions throughout the whole cell. As long as it has enough amount of potassium positive anions, or cations, the cell remains neutral in millivoltage. But as law of diffusion takes place, potassium moves outside the cell slowly but surely. There is less potassium and more anions remaining in the cell. This results to a high negative charge inside the cell. Now, as I said, there's an extreme amount of potassium inside the cell. It is not nowhere near becoming equilibrium concentration between outside and inside the cell for the potassium law of diffusion to stop and equal in a net movement of potassium. So why does potassium stop once the cell reaches negative 90 millivolts? Well, what happens is like this. Potassium starts leaking out, becoming negative 10, 20, 30, 40, 90 millivolts. Once it becomes negative 90 millivolts, what happens is there becomes an extreme negativity inside the cell, and that has an electrostatic pull on any positive anions nearby. So not only does potassium stop leaking outside of the cell because of this pull, any potassiums outside the cell get pulled back inside the cell due to the electromagnet pull. So now what happens is you have two forces at bay. Law of diffusion forcing the concentration of potassium to go to lower, which is outside the cell, and the electrostatic pull, which is pulling the potassium back inside the cell. Where do the forces balance out? Well, our friend Nernst figured out an equation to find out. And according to the equation, it's at negative 90 millivolts, which means once the cell becomes negative 90 millivolts, the amount of negativity is the exact amount of force as the law of diffusion, and therefore it causes no net movement of potassium. So there we go. Now our inside the cell is negative 90. So the question remains, why do most textbooks say that the resting membrane potential is negative 70? Well, the answer is the third factor that we have for our resting memory potential, which is our sodium leaky channels. So besides the potassium leaking out, we have sodium leaky channels leaking in. Now, once sodium channels leak in, even though it's not a lot, there is still a decent amount of sodium leaking in, and the sodium are positive cations. They leak into the cell, and they stop when the cell goes into a positive or reaches a negative 70 millivolts. At negative 70 millivolts, you have no net movement of potassium cations and no net movement of sodium ions, and the cell reaches its resting memory potential. Very good. Now that we know how resting memory potential works and the three factors that make resting memory potential negative 70 millivolts, the next thing we're going to go on is to the synaptic transmission of neurons. So, simply put, what is the synaptic transmission? It's when a neuron gets a message from the brain cortex. It needs to relay the message over to the next neuron. Now, what happens is, between one neuron to another one, there's a gap. This gap is crossed via the synaptic gap, which are transmissions crossing one neuron to the next one, relaying over the message. So now we're going to find out how this works exactly. So, first thing, as we said, the first neuron sends chemical transmitters to the next neuron. Now, there's two types of chemical transmissions. One depolarizes the next neuron, which gets it to fire, which means it gets it to negative 55, and this is called EPSP, excitatory postsynaptic potential. Great, it gets the neuron excited, and it releases a potential of negative 55 and gets it fired. So let's deal with this first. What happens? So let's look at our blue chemical messenger. The most common chemical messenger is glutamate. Glutamate is sent from a neuron to the next neuron to relay the message. What happens? Glutamate is sent and is received by our neuron via its receptors, which are ligand gated ion channels. Now, as soon as glutamate binds on to these channels, or rather before glutamate binds on, we can see in our depiction here that the channels are closed. It's not the best drawing, but the channels are closed. Once glutamate binds on, then the channels open. Once they open, they let a certain ion come into the cell. And what are those ions? So it depends. There's two types of opening. One allows only sodium in, the other one allows sodium and calcium in, but in general, it allows either sodium or calcium or both. Now, what does that do? So you can easily tell that adding cations into a cell would make it more positive. Great. And making it more positive would help us reach our threshold. Now, on the other hand, we have another type of messenger of a message coming from the previous neuron. What is that? That is the process of IPSP, which means inhibitory postsynaptic potential. Now, what does that mean exactly? It means like this. Excitatory gets the cell excited, which would fire it off. Inhibitory means it would inhibit it and get it even more negative. What is the process of that? So, one of the most common inhibitory chemical messengers is GABA. GABA comes in from the previous cell, and before it comes in, the ligand-gated ion channel is closed. As it attaches, it opens in, but instead it lets in the opposite types of ions. Who does it let in? Chloride, negative chloride ions, to make the cell more negative than it is. Now, the question is asked, if we have excitatory postsynaptic potential bringing in positive ions, and then we have inhibitory bringing in negative ions, we're not going to get anywhere. So how do we get somewhere? How do we get the cell to fire specifically? Well, very simple. The step to getting the cell to fire to 55, you can't have it at the same time negative 90 and negative 55. So there has to be either one. And that's exactly how it works. If you take a chart, and we're right now at resting membrane potential of negative 70. Excitatory, small excitatory postsynaptic potential only get the cell to, let's say, 65. And then another excitatory gets it up to 60. We're not at the threshold potential yet. And then let's say we have inhibitory, getting it back down to 80 and maybe 90. And then we go back to 70. Once we have enough of EPSPs, eventually we hit negative 55, which is our threshold potential, and then we get going. So, before we move on, quick thing we need to cover is how does one neuron send its chemical messages to the next neuron? There's two ways. First one is called temporal summation, which means that one neuron sends excitatory message to the next neuron once, twice, 3 times, 4 times in a rapid one after another, rapidly, and that is enough to excite it to our threshold potential. The next way is spatial summation, which is rather than one neuron sending multiple messages, it's multiple neurons sending one message or 2 messages. And when enough chemical messages hit our neuron receptors, it eventually becomes positive enough because enough sodium or calcium goes in. It becomes positive enough, and we hit our negative 55 threshold potential. Now, we've been speaking about negative 55 threshold potential for this whole video. What happens at negative 55 millivolts? Well, it's a process of action potential firing, which we will be discussing now. So, let's take this picture. This is the part of the neuron after the dendrites. So we received our chemical message in the dendrites and the soma part of the neuron. Now we go on to the axon hillock, and all the way until the end is the axal terminal. What do we have here? So it's not depicted in the picture, but we have marked in purple is our ion channels that will be letting sodium in. And why will be letting sodium in? Well, let's find out. So what happens is right here at the beginning of the axon hillock, which is called the trigger zone, is that these are not ligand-gated ion channels, and they're not leaky channels. These are voltage-gated ion channels, which mean these channels will not open unless our cell hits a certain voltage. That voltage being negative 55 millivolts. So once we hit the threshold potential of negative 55 millivolts, the first few sodium voltage gated ion channels open up since we reach the threshold. And what happens is sodium starts flowing in and rushing in the cell, making it more and more positive. Now, it goes on in a wave from left to right, from the beginning of the cell to the end of the cell. Once the first few channels start becoming open, the cell becomes more positive. Then the next couple channels start opening up, the cell becomes even more positive. Then the next few channels start opening up, and the cell becomes so positive until it reaches positive or over positive 30 millivolts. Now, why is positive 30 millivolts important? Because that is our next threshold for continuing the firing. At the end of the cell, which is the axon terminal, we have, again, voltage-gated ion channels. And these specifically open up at positive 30 millivolts. And once our cell reaches positive 30 millivolts, what opens up? An ion channel allowing calcium to flow in. What happens when calcium flows into the axon terminal, you might ask? Well, the answer is very simple. You see these three circles that we have here? These are vesicles carrying chemical messengers. Now, at the end of the vesicle, you see there's a blue dot drawn. That is a protein. At the end of the membrane of the terminal, there's another protein. What calcium does is combines the two proteins to work together and join up. So what happens is once calcium is released, the vesicle of the chemical messenger is binded to the membrane terminal, which then it becomes part of the membrane terminal or terminal membrane. Once it becomes part of it, the next step of the cell is to release the chemical messengers out of our neurons onto the next neuron's receptors and begin the process all over again for the next neuron. Great. So now we learned that there's a resting membrane potential. Once the chemical messengers get binded to the receptors, we get our millivoltage to the threshold potential. Next, our axon or our neuron fires all the way down to our calcium channels and releases our chemical messengers to the next neuron. Great, but it's not over yet. Why? Because if you noticed, for our calcium ions to come into the cell, our cell is probably at positive 30, 40, 50 millivolts. Now we know our resting membrane potential is negative 70. So the cell has to polarize, basically calm down, get its millivoltage back negative. How does the cell do so? Well, this process is called repolarization. Now, what happens in repolarization? If we go back, what happens is, as the sodium channels open because the cell became negative 55, at the same time, simultaneously, potassium channels open. Now, potassium channels open outwards, which means potassium starts leaking out. Now, if potassium starts leaking out, the less positive cations we have in the cell, the more negative the cell becomes. So the more potassium that leaves the cell, the more negative the cell becomes. Then the next potassium channels open, and the more negative the cell becomes, until the cell becomes negative 90 millivolts. And once the cell becomes negative 90 millivolts, it goes back to the calcium channels, which need about negative 90 millivolts to close. And once they close, the cell is back to negative 90 millivolts. And then we know that eventually it diffuses out and comes back to negative 70 millivolts, resting membrane potential. Now, what I want you to notice that I've pictured it in a way that first the sodium channels open, then the calcium channels open, and then the chemicals are transmitted to the next neuron. Then the potassium open, make the cell negative, and then they close the calcium channels. Well, it is true, but not as you might think. This happens simultaneously. Once the sodium channels open from left to right and are making the direction of the cell going rightwards, positive 30 millivolts, right behind them the potassium channels are opening up and making the cell negative 90 right behind them. So there's a wave of positivity, positive 30 moving to the right, opening the channels, the calcium channels, and allowing the the chemical transmitters to go to the next cell. And right behind them, there's a wave of negativity of negative 90 millivolts following them and closing back the calcium channels. So that's how it works in a real neuron. Now, we've covered everything. Let's go on a quick recap. And before the quick recap, as we can see, I didn't mention it, but we have something called myelin sheaths between the axon hillock and the axon terminal. And not all cells have a myelin sheath, but basically what a myelin sheath does is the process of sodium cations moving throughout the cell and making the cell more and more positive, and at the same time, right behind them, the potassium ions move throughout the cell, making it more and more negative, goes relatively slowly for a neuron that doesn't have myelin sheaths. But neurons that have the myelin sheaths, it goes much, much quicker. In real life, I saw that cells who don't have the myelin sheaths have a 5 millisecond response time, while cells or neuron cells that have the myelin sheaths have about a 1 millisecond response time. So even though it's an extreme amount, but for neurons, having the myelin sheaths makes it much, much quicker. Now, as a recap, we have our neuron sending a chemical message to this neuron. It receives it on the dendrites of the neuron and the soma, which is the cell body. The neuron becomes excited and becomes negative 55 millivoltage. That activates our sodium channels on the axon body, slowly but surely making a positive wave until we get to the axon terminal, releasing the calcium ions, and then going to the vesicle chemical messengers, binding to the terminal of the membrane and releasing the chemical messengers to the next neuron. So now we've covered everything, and I hope you enjoyed. I appreciate everyone for being here. Thank you, and thank you very much, Professor, also for the subjects you allowed us to learn. It was very fun learning this and making this, and I really appreciate everything. Have a good night and take care.